Welcome to the Explainer. Today we're opening up the scientist's toolkit to understand a truly fundamental process, plasmid extraction. Okay, let's dive right in. We'll start with what a plasmid actually is and why it's so important. Then we'll walk through how scientists crack open cells to get it, how they clean it all up, and how this whole process is different from other kinds of DNA extraction. And we'll finish up with a handy checklist for getting it right every time. So first up, what exactly is this genetic tool we're trying to isolate? All right, so what are we even talking about? A plasmid is basically a tiny circular piece of DNA. It's not part of the main chromosome you'd find in a cell, like in bacteria. And the really cool thing, it can copy itself completely on its own. That's why you can really think of them as the USB drives of genetics. I mean, this ability to be easily copied and passed around, well, it makes them absolutely essential tools for molecular biologists. Scientists can load them up with new genetic info and then pop them into other cells, kind of like plugging a USB drive into a computer to move files around. And that is the central challenge, right? It's a really delicate and precise process to get these plasmids out without damaging them or getting the sample all gunked up with other cellular junk. Okay, so how do we actually get these plasmids out of the cell? Let's get into the nitty-gritty of the extraction phase. The main idea here is to be gentle. You've got to carefully break open that bacterial cell, just enough to let the small circular plasmids sneak out, all while leaving that big tangled mess of chromosomal DNA behind. It's kind of like trying to get a key out of a locked box without scratching the key or spilling everything else inside. Now, this is where scientists have to make a choice, and it's a classic trade-off. On one hand, you've got alkaline lysis. It's fast, it's cheap, and it's pretty much the workhorse of many labs. The downside? The DNA you get isn't always super pure. Then there's the boiling method. It's incredibly simple and quick. You literally just boil the cells. But that heat can actually damage the plasmid DNA. So you'd only use that if you're in a real hurry and purity isn't your top concern. Then we have the more advanced options. You've got your kit-based methods, which are super popular, they're really user-friendly, and give you high-purity DNA. The trade-off? They cost more, and you might lose a little bit of your plasmid along the way. And for the perfectionists, there's the enzyme lysis method. This gives you exceptionally pure DNA, but it's complex, it's slow, and yep, it's gonna cost you. So an extracted plasmid is still pretty crude. It's all mixed in with cellular debris. The next step is really to polish the tool. Right, so just getting the plasmid out isn't enough. It's still floating around with a bunch of unwanted junk, proteins, salts, you name it. So first, you gotta remove all those contaminants. A common way is using these little spin columns that grab the DNA and let you wash everything else away. Next, you need to concentrate the DNA. And for that, ethanol is your best friend. And finally, the most critical step, you have to assess the quality. Did it actually work? And quality control is everything. You can't just cross your fingers and hope for the best. Is the tool you just spent all this time making even pure enough to use? Because if it's not, your next experiment is basically doomed before it even starts. And when we talk about purity, there is a magic number that every scientist is looking for. And that magic number is 1.8. It comes from something called the A260/A280 ratio. Now, it sounds complicated, but the idea is simple. A machine shoots light through your sample. DNA absorbs the most light at a wavelength of 260 nanometers, while protein absorbs the most at 280. If the ratio of those two is close to 1.8, bingo, you've got really clean DNA. But that one ratio isn't the whole story. For a more complete picture, scientists have other tricks up their sleeve. A qubit assay, for instance, uses special fluorescent dyes that only stick to DNA, so it gives you a super precise measurement of how much you actually have. And then there's gel electrophoresis. This lets you literally see your DNA, so you can confirm it's the right size and that it hasn't been chewed up or degraded. Okay, so this is a really important distinction to make. Getting a plasmid out of a cell is not the same thing as getting all the DNA out of a cell. They're two totally different ballgames. This table really lays it all out. Plasmid extraction is like a targeted mission. You're going in for one small, specific tool. The methods are gentler, it's pretty quick, and sometimes good enough purity is, well, good enough. But genomic DNA extraction, that's a whole different beast. You're trying to harvest the entire genetic blueprint. This means the process is way more aggressive, it's more complex, and you need exceptionally high purity for things like sequencing a whole genome. So, to wrap things up, let's run through a final checklist. These are the things scientists keep in mind to make sure they get great results every single time. Every single one of these points is a tiny detail that can make or break an experiment. You've got to start with the right culture conditions, then be super careful with temperature and time when you're breaking the cells open. You have to be almost paranoid about contamination from enzymes that can just chew up your sample. And, of course, choosing the right purification method is key. Finally, storage. You stick it in a freezer at minus 20 degrees Celsius or colder, and try not to thaw and refreeze it a bunch. And that's why this is such a big deal. Plasmid extraction really is an indispensable technique. It's the very first step for so many things: gene cloning, getting cells to produce specific proteins, even developing brand new genetic therapies. Mastering this one process just unlocks a world of possibilities. Which leads to the big question, doesn't it? Now that we have the tools to isolate and manipulate the very code of life, what should we write with it? That's the question that drives science forward. And it all starts with a perfect extraction. Thanks for watching The Explainer.